December 2nd, Yuancheng Lu from Dr. Sinclair's lab published a paper entitled Reprogramming to Recover Youthful Epigenetic Information and Restore Vision. Today I will go through some of the highlights on this paper. The ideas behind the paper are based on Dr. Sinclair's theory of loss of epigenetic information in aging and recovering that information to restore tissues to an earlier age. They decided to work on the eye, and in particular the optic nerve, which is actually part of the brain. In the experiments, they show that by applying 3 of the Yamanaka factors, they restored youthful DNA methylation patterns and promoted nerve cell regeneration after injury and vision loss from glaucoma. As nerve cells lose their ability to regrow shortly after birth, so showing that they can be rejuvenated to the point where they can regrow and recover from injury and disease is really exciting. Let's have a look at some of the key points in the study. The first test they did was to reset the epigenome without erasing the cell identity. You may recall that this is a similar approach to the ERA technology from Dr. Sebastiano and Turn Bio. What they did was to express 3 of the 4 Yamanaka factors in the cells of the old mice. They saw that after introducing the Yamanaka factors, the messenger RNA profile of the cell resembled that of a younger cell without losing the cell identity or causing cancer. Let me briefly discuss the Yamanaka factors. These are genes which, when expressed, can take a normal cell back to being a pluripotent stem cell, or a cell which has the ability to become any kind of cell. However, this would mean that the cell has lost its identity, which for this case they did not want. They wanted the cell to remain the same kind of cell it is, a nerve cell, but to go back to being a younger version of itself. We will talk about how they do this in a minute. As shown, there are 4 Yamanaka factors, but c-Myc causes cancer, so they removed this. The remaining 3 are OCT3-4, SOX2 and KLF4, or OSK. What they found was that the remaining 3 were sufficient to cause the rejuvenation. The next test was to show the safety of expressing OSK in vivo. For the experiment, they needed to have OSK factors expressed for a specific time, and not always on. They did this by infecting the mouse with a harmless virus which has been engineered to contain the OSK genes. However, the genes were not being expressed, that is to say, they were off. The genes were engineered in such a way that in the presence of doxycycline, they would become active. So this allows the experimenters to control when the gene is active by either feeding the mouse or injecting it with doxycycline. Next, they induced an injury in the optic nerve and then caused the OSK factors to be expressed to see if the nerve would recover. Here we can see the timeline. First, inject the virus with the OSK genes into the eye. 2 weeks later, damage the optic nerve. 2 weeks after that, they injected a substance which would stain the optic nerve and so show whether there had been any recovery. Here we can see the result. In this picture, the light areas are active nerve cells, and we can see that with OSK expression, they have grown beyond the crush point, whereas without it, there has been little to no recovery. This chart shows that when the 3 factors were expressed, the number of nerve cells and how far they grew was much larger and longer. A couple of other interesting points that came out with the experiment. The first of these was that after the crush injury, the methylation pattern of the retinal cells, the RGCs, became older. But when OSK was applied, you can see here in this diagram that the methylation returned to much closer to being earlier in the model. Another point was that when they turned off the genes that actually perform the methylation process, they did not see the same results, showing that the methylation is a required part of the process. 2 other experiments were performed. The first of these was to see if mice with glaucoma could recover sight, glaucoma being a common reason for loss of sight in older organisms, and using a technique to restore vision in old mice. In this, they treated 3 and 11-month-old mice with OSK, and they saw that vision was improved in those with the OSK treatment.
We could put mice in front of a screen where lines are moving, and if a mouse is blind, it will just sit there staring. But after we treated the mice, what happened was we saw that they were starting to move their head like this in response to those lines, showing that they had got their vision back. And that, as far as we know, is the first time that's ever been possible.
Here we can compare the methylation patterns for the young, the old, and the OSK-enabled mice, and we can see that the pattern of the mice which has been treated with OSK more resembles that of the younger mouse than the older one. I agree with the point raised in the discussion. Perhaps the most interesting question is where the information as to how a cell becomes young again is encoded. When the OSK factors are expressed, how does the cell know which site should be methylated and which not? A very hopeful and interesting study. Looking forward to more progress on this technology.